significant amount of time, so I apologize for that. I did post, and you'll see, like, for your guys' student view, it shows up here, unknown explanations, a link for you guys to be able to remember what you were supposed to type up so you could submit it. And you don't have to email it to me, and I don't get 1,000 emails. I got like one email last night, and one person that noticed after I had posted it. But notice I put a deadline on it too, that I found out that's why it shows up over here on this page. Figured out how to do that. But so, since I didn't go over a good example for you guys and didn't provide one in a timely fashion, I'm giving you more time. So when you click on the link, which is also under Module 8, you can either click on this to view it right on the page, or you can download this document. And so even if you already turned it in, you can resubmit, by the way. So your explanation, so this is one of the harder ones, and that's teeny tiny. But you're going to need, obviously, your results if you want to go back over and check and make sure. And you can get a copy of the unknown report. It's on Canvas under Lab 8 as well, right? That's when we started all the work on your unknown. So if you want, I'll make you a photocopy, right, of your report. Does anybody not want a photocopy of their report today? Okay. So while you guys are doing the activity, I'm going to go to the copy machine, and I'm going to make a copy of your report so that you can look back over it and you can see if you did like I did in these explanations. So like if someone had the result for their phenol red broth, that the media was yellow and there was no bubble in the tube, and so your result was A-, right? This is the explanation. And if you read through this, this is an example of the explanation, right? See how it's a paragraph, how I thought, you know, you guys, there's no way you're going to fit them on your unknown reports if you really did what you're supposed to do. If you read through each one of these, I did all 4 of the possibilities, right? Check. I answered all of these questions in that paragraph. Every single one of them. And so for your paragraphs, for each one, you need to answer all those questions, right? In a paragraph that makes sense so that you guys know, and I know you know this stuff, right? And you understand how these tests work. That make sense to you guys? So that's for everything that was in a tube or on a plate, right? Every test we ran that we had all those questions, the only thing you don't have to write a paragraph for is obviously your stains and the motility tube, right? I don't ask you guys to really write a paragraph on that one. I don't ask you a bunch of questions. Right? So it basically starts, where's the report? Right, so you have to write an explanation for anaerobic growth, right? Why is it you picked the one you picked? That's a relatively short explanation, but still you probably need more than the 2 lines that are here, right? Phenol red I did for you, right? You could paste and copy mine, I don't care, right? But check and make sure I didn't miss anything though, right? Citrate you got to do, catalase, right? All these pages, see how it says, using the information you provided in the above questions, explain in detail what your organism can or can't do based on its results for this test. For every single one of them, right? Huh? No, this is part of the report. I'm giving you guys more time to work on it. This is a 50-point report. You could write it. If I can't read it and you didn't answer all the questions, you will lose points. So it's your choice. I'm giving you guys till the 22nd that you can submit it electronically, right, through Canvas, written up. Okay. So the way the link works is, that's why I did it in my student view. So you notice right here it says submit assignment. So you can upload a file by clicking browse and finding the file on your computer, or you can paste and copy into a text entry. So either or, whatever works for you. It won't let you do one or the other. It won't let you do both, right? And if you've already submitted, you can submit again, right? So if you work on it and you submit it and then you decide, oh, I want to work on it some more, you can resubmit, right, a newer version, okay? Up until the 22nd at 8 a.m. At that point, it's done, okay? So we're going to go over some of this stuff, right, today. So one other change that I made this morning because I realized I still didn't calculate the stuff right. It was screwing up the grade book. And I didn't go over it last time, is how grades are calculated. So this is my test student, so this isn't anybody's real grades. But you could put in, you know, after today, what you think you could get on the final, and it'll calculate accurately now what your average is. You have to hover over the points, and it'll tell you your percentage. So originally I had made the staining your quiz 8, but I had to get rid of that because there was no way I could add it into the quiz section or to do the drop quizzes. So I just turned it into bonus, right? So you'll notice that it's referred to as bonus now. So these are all bonus, and that's why there's 0 points, but you could get up to 2 points, right, for those. If for some reason you haven't got those done, I don't know how you handed in your unknown report today, unless you looked at them otherwise. But you'll notice, like for this one, it's Xing out some of these homeworks. That's because we had 14 overall homeworks. Only your 10 highest ones will count. So Canvas automatically deletes your lowest grades, right? Grays them out. They don't get added in to your homework. So when you look at your homework, right, if it's 100%, which right now I just entered everybody's homework in, right? If you have 100%, if it says 50 out of 50, you really do have 100%, okay? Because they're 5 points apiece, and we keep 10 of them. So as far as the quizzes go, right, I just made what I checked this morning, that made that a bonus quiz worth 10 points. So in the quiz total, so another bonus, what I mean by that is basically an extra one, right? And remember, serial dilutions I reopened, right? If you hadn't done that, or you didn't have your perfect score, you might want to go back and redo serial dilutions, right, and get your points for that, as well as I reopened quiz 6, right? So these are almost like bonus quizzes, right, or extra quizzes. So we end up with, let's see, 1, 2, 3, 4, 5, 6, 7, say that's 8, 9, 10, 11, 12, 13. So you get to drop 3 quizzes, right, because only the 10 highest count. So once I put in your quiz grade from today, right, that quiz grade in here will be accurate, right? It'll be 100 out of 100 because I will have entered all your quiz grades at this point. And then staining, as I said, is bonus. That's why it's not adding up here, but you can hover over and see what the total was for that, and it should hopefully be 10, right, points, which would be great, right? And then you have the possible of a total of 7 points from exam 1. So you could have potentially 15 bonus points. If you helped out with the expo, which some of you guys did, there's going to be one additional thing where you can add in bonus, and that's you can get 5 more extra bonus points for helping out with that. Okay, because the max is 20 for this class. You can't get more than 20. Okay. Any questions on that? Okay. So you'll know by today, other than, again, you can kind of guesstimate what you're going to get on your unknown report, right, which is worth 50 points. It's a lot of points. And I will have them graded and handed back the day of your final, which for you guys is December 5th, right? In here, it's a Monday at 11 o'clock. Where's December? December 5th, it's a Monday, in here at 11 o'clock. There's no hands-on practical, right? No aseptic transfer or anything like that. No focusing on microscope. Just, you'll have to look at microscopes, right? And you'll have to look at media and answer questions and know your terms. So in preparation for that, how do I get back to my class? Because we're going to actually do a lab today, believe it or not. The last one. It's kind of fun. But in the exam module, there's the worksheet that we're going to go over, right, that I gave you 10 bonus points, 10 quiz bonus points, right, extra 10 points for for filling that out for today. There's the practice staining ID. I don't remember if I opened this one up before for you guys before. There's the other practice one. Plus I have a practice quiz for today's lab that you guys can do. Remember, practice quizzes don't count. You can do them as many times as you want. They tell you the answers, right? And then I have the PowerPoints that I'm going to use today. So, speaking of today, unknown stains. So these are actually your guys' pictures. Some of you guys, not all of them, right? And yeah, it's actually coming out pretty good up there. I'm pretty happy with that. I wasn't sure how it was going to project. Okay, so color-wise, what colors are you seeing here? Purple. Right, so what are you guys calling this? Like a purple? Okay. What color are these? Blue. Okay. So what stain might this be? Those are endospores that are staining purple. But this stain was for having blue cells. The cells are blue, right? And the endospores are actually coming up purple. What would give you a blue result? Acid fast. And this person obviously has a spore former, and we've never really done this before because remember I gave you guys old cultures, right? They were 48 hours old, right? So we actually had some with spores, endospores, and I've never seen this before because we've never done that before with spore formers at the spore stage, and probably explains why I actually have done in the past and some of my students got the wrong results. Nobody in this class has an acid-fast organism, y'all. No one, right? We can't work with mycobacteria. They're pathogenic, right? So you guys looked at acid-fast under the microscope, hopefully, the Mycobacteria tuberculosis, and you would see like a purplish pink color like you're seeing here for the endospores. So that was kind of an interesting result that I wanted to share with you guys. This one over here, on the other hand, is the endospore stain. And so these bluish, supposed to be green, right? These aquamarine, you could say, structures you see here are the spores, the kind of lightly pink, almost purplish even, because the color's off on this. This is an endospore stain with endospores. So what is this stain? Well, what's this one then? If this one's negative, what's this one? Yeah, this one's the capsule. This one's the capsule. And again, the cells didn't stain all that well, and the endospores didn't either, right? You see the endospore inside the cell. But yeah, this space, this gap between the background and the cell, right? That's the capsule. This is a capsule positive. And again, look, you can notice at this one, right? We definitely see it, I think more so in this picture, right? It's rod shaped, but they're definitely forming chains, right? And that's why I have you guys do the negative stain, which is over here, where again, you can see that they're chains, but you really can't even see the cells, right? The endospores are the ones that are really shining quite bright in this picture, right? So this is a negative stain, right, where the endospores are actually quite prominent, more so than the cells. So this was an interesting one. I forget whose this was. There was someone in this class. So this one is no doubt what? Those were yours? What is this one? This is endospore. And again, see the endospores, and you can really see the pink cells in this one. So this is an endospore positive. What's this? That's capsule, right? Because you can see the cells are stained and you can see that gap, so it's capsule positive. Again, rods, we got a spore former. What's probably this one? Gram positive, right? And again, you can kind of see, right, the spores that didn't get stained, right? But that's gram positive. And then this one is, this is acid fast, and the result is negative. But again, if you look closely, you can see little pink spores in there. This one, another spore former, huh? This is the spore stain, right? This is a really good one. I might steal this one for the test. This slide. Okay, what's this? Capsule. What's the result? Positive. Oops, sorry. I went in different order, right? This is acid fast. Again, mostly you can just see the blue on this one. You can really see the pink ones. So this one's capsule. And notice, you know what these are? These little things? They're the spores. They're the spores. Right? But you can see a little bit the spores inside here. But you can definitely see that halo. And then this is gram what? Positive. So what about this one? You think so? Yeah, this is purple right here, right? So that's probably the gram stain picture, right? In case somebody didn't label their slides, right? This is definitely purple. Everyone agree with me on that? This is purple, right? I asked what this one was, right? And you guys said gram negative, right? Which, because it's pink, right? But you've got a purple one over here. And if you look at our sheet, by the way, y'all, all cocci were gram positive. That was the only choices. We don't have any gram negative cocci. And these are definitely cocci, right? So this is probably the gram stain. This is probably negative stain. This one's blue, so it's probably acid fast. So what are we left with? Endospore. It's endospore, but it's endospore negative, right? You don't see the little green things, right? This one's gram positive. This one's acid fast negative. This is a negative stain. You can really see the cocci in clusters, right? What about this one? It's kind of what color? Bluish, so it's probably acid fast, and the result is negative for everybody, right? This one, capsule, and again I zoomed in on this one so you could see that it is a rod, right? And it does have a capsule, right? You see the halo? Okay, so now we got two pink slides down here. Right, one's endospore negative and one is gram negative.
How do you know the difference?
You'd have to label your slides. Those really are two different pictures, y'all. But they don't really look it, do they? Okay, so what about this one? This one's really hard to tell the shape, right? Because they're so tiny. These are actually little tiny rods. Do you see that?
What?
Yeah, little tiny rods. And what stain is this?
Negative.
Negative, right? And so again, look, I actually put two different ones here. So these are what stain? So you can see the cell, you see this gap around the cell, and you see a background. You see the cell, you see a gap around it, and you see the stained background. Capsule, and it's positive. Okay, now we got two pink slides again, right? So one of them's probably gram negative, the other one is endospore negative, and you know, this one has some clues. Because do you see these green, right? Little bits of green? That's excess dye that didn't get washed off that slide, right? And some people sometimes think that theirs is endospore positive. Your endospores, as you guys seen in those other pictures, are going to be smaller than your cells, right? These, you see how big they are? Like big blobs of green compared to the cells? That's just leftover dye. So it is endospore negative, right? This is my favorite capsule one, huh? Isn't that beautiful? Nice big rods, nice big capsule, capsule positive. So, what about these other pictures? What do we see here? What color does that look like to you guys? Yeah, these guys are really light purple, but these almost look blue, right? They're so dark. These are definitely what color? Blue. So this is definitely probably what? Acid fast and acid fast negative. This is probably gram, right? So therefore, what's this one? Endospore. And that's actually pretty much all you see. And do you see how they have a pretty defined shape to them? Right? All these bluish things, those are all endospores. This one was just loaded with endospores. This was a much older culture. And these are just some of my favorite because they just look like art. Right? Didn't they? Right? So definitely bacillus in chains, right? You can really see that. That's a what stain? That's negative. What's this one? Gram positive. Yep. Here's another really pretty one. That's endospore. You can really see the endospores and a whole bunch of packed in red cells. What's this one? Capsule, and it's positive. And so I'll post that for you guys. But then there's also those practice quizzes you can go back over. And for the test, what I'm going to have is I'm going to have an acid-fast negative and positive, and the question will say, tell me which one's positive or which one's negative, microscope A or B. Same thing for all the stains, right? I'll have a positive and a negative, right? Or even a distractor, and you have to tell me which one is what I'm looking for, right? So you need to be able to identify stain results. I'll tell you which stain you're looking at, but if you guys can do like you just did right now, you're golden, right? You even could figure out what stain you were looking at based on the colors, right? Okay? Any questions on that? All right, so let's go over unknown report. So, unknown report highlights. So the genus and species should always be italicized or underlined. I'm not a real stickler for this, right, when you guys are writing, but just remember that, right, in the future, you should always italicize when you're typing and underline when you're writing it. The word bacillus, a lot of us tend not to use because, again, it is also a genus name, right? Some of you guys, your organism is Bacillus cereus or subtilis or megatherium. So we typically say rod, right, and they have this overall shape. And then we have cocci, right, and they can have different arrangements. So you still need to know shape and arrangement, right? But remember, in our lab, it's pretty simple as far as our unknowns, right? They're either cocci or rod, but we have a couple of different arrangements. Gram should actually always be capitalized, right? It's a name of the guy, it's a name of a procedure. If you lowercase it, it could be confused with gram, which is a unit of weight measurement, right? And you don't want to do that. Remember, the gram staining procedure is probably one of the most widely used procedures, right? It's not the easiest one. You guys have hopefully gotten a lot better at it. And it really helps us see understand what the potential ultrastructure of the membrane and cell walls are like. So remember, the gram-positive have that really thick peptidoglycan layer, traps in the crystal violet iodine, where the gram-negatives have that thin layer, if they have it at all, and typically have that outer membrane. So they decolorize and they get stained pink. And so, especially for your guys' gram stains, right, that if you did that early enough, you could actually divide the possibilities of unknowns in half, right? And that is the way pretty much for most investigations of unknown bacteria, right? Knowing whether it's gram-positive or gram-negative narrows your possibilities in half. And then remember, our selective medias helped also confirm those results, right? They either grew or didn't grow depending on the selective media for the CNA and EMB plates. So gram-positive, right, purple, gram-negative, pink, and we did positive controls, right, because it's a very technique-sensitive result if you don't do it properly. We did negative stains. You guys mostly did negative stains because that's probably the best one for arrangement and shape, along with the capsule stains sometimes. But again, that one, there's some difficulties with that procedure. So these guys are real helpful. Endospore to help prove that if you were seeing maybe in those other stains like the gram stain or the acid fast, if you do the spore stain and you really do get those awkward light colored spores, and you know that those are in fact spores that you're looking at. For the acid fast, remember all of ours were negative. They were a bluish color because we used the counterstain methylene blue. For acid fast, remember they're going to be that purplish fuchsia pink color. And they're rods, right? They're commonly abbreviated acid-fast bacilli, but they tend to clump together because of that mycolic acid in their cell walls, right? It's hard to kind of get them apart and see that they're rods. So, again, no arrangement terms, right? So this is two bacilli together, so that'd be diplobacillus. So, being capsule negative is very rare if you look at the sheet, even in our class, right? There's only one that really should have come up negative. Endospore positive is kind of rare, although it didn't seem like that in this class, right? Because I gave out a lot of spore producers. And of course, we don't even work with the acid-fast positive, and they are rare in general too, in nature. Thank goodness, right? Because most of them are highly pathogenic. So in food manufacturing, they want to make sure that Clostridium botulism endospores aren't in, because it produces a toxin when grown in canned foods. So one of the things that a microbiologist would do in the food industry even would be to do an endospore stain, right, on a product to see if there were endospores present in that product. So, you know, these are valuable techniques that you guys have learned. Whether you will actually really use them later on in life depends the career path you choose, right? But at least you have hopefully a pretty good lab foundation. Organisms that are motile are usually propelled by the means of flagella. In the case of our class, they are propelled by flagella. If you had E. coli, he's actually completely covered in flagella and moved out in his motility media. So remember, we use the reagent TTC to be able to see where the growth is, because it turns red in the presence of the growth. In the case of a negative one, if you stab it into the tube, remember, it doesn't move because it's non-motile. It stays a straight line. So again, you'll have these tubes out, and you'll have to tell me what's the result. Is it positive? Is it negative? Right? On a set of tubes. For the fluorothioglykylate media, right, remember we have an oxygen zone versus a non-oxygen zone. And based on that growth pattern, you can determine the classification of your organism and if it's utilizing oxygen. Which is the case for all of ours. They're utilizing oxygen, right? But some of them can grow anaerobically additionally to aerobically. And those were which ones? That, yeah, facultative. So some of you guys had growth all throughout your tube. It was not an aerotolerant anaerobe because the one we had is pathogen, so we had to get rid of it. But it's also hard for most of you guys to see because of the age of the tube. You would look at this the next day, even though we refrigerate them for you guys. It's kind of hard to see that you actually have more growth at the top than you do at the bottom, especially since the way we had you guys inoculate them. I might actually try and do a stab next semester and see how they come out. But I had some examples, you guys remember, where you could see it looked just like this picture, where you had less growth in the anaerobic zone and more growth in the aerobic. And then the other choice you could have had is a strict aerobe, where it only grew in the oxygen zone, right? There was no growth in the no oxygen zone. All of these tubes, right, these growth patterns you need to be able to identify for the test, right, and understand what's going on. Although I won't have tubes, I'll probably just have pictures because it's too hard to have them prepared for a Monday lab. I'm not coming in on Sunday. Not to mention the police give me a hard time about coming in on Sunday. Like, do you really think I want to be here, though? Okay. So, phenol red block tubes, we will definitely have each one of these for you guys to identify. Right, so you should know the code that we use for these. I think you guys probably have a pretty good grasp. I hope the activity we did helped you with that. And then reading over the explanations that I have for each one of these, I think will definitely help you. I want to check in with a couple of you guys because you're supposed to re-incubate this phenol red, and phenol red. And I don't know if you looked at it after it was re-incubated, right? You might need to check that to confirm your results. Suzanne, I think you might be one of those ones. Huh? Did you ever check it? Did it turn pink? Right, so it did turn pink? Okay, all right. See, I knew there was someone in this class. Good. All right, so you got that result. Good. I just wanted to make sure. Okay, so citrate permease, remember, if your organism has that, then it can bring citrate in and use that one carbon source that's in the media, and then it'll also utilize the nitrogen, which changes the pH, and those tubes turn what color? Blue. They go from green to blue because the bromenthal blue, right? And that means that your organism has citrate permease, and it even grew, right? If yours doesn't have citrate permease, right, what happened to it? It died even. It died, right? And because it died and it didn't grow, you didn't get the production of the base that turned the tube blue, it stayed what color? Green. So again, you'll have tubes of that. You'll have to be able to identify that result. So catalase test. This is an enzyme that detoxifies, right? Hydrogen peroxide, that was the reagent that we use. When you add hydrogen peroxide to an organism that has catalase, it breaks it down into water and gaseous oxygen. So what happens when we put the hydrogen peroxide on your cells? It bubbles, right? What's in those bubbles? Oxygen gas is in those bubbles, right? What type of organisms need this? Organisms that do aerobic respiration, right? Which are strict aerobes like ourselves. And who else? Facultative anaerobes, because they do aerobic respiration too, right? When oxygen is available. So they're using an electron transport chain. They have oxygen they're using as an electron acceptor. Electrons can jump ship from that train, remember, and form toxic forms of oxygen. What's the most damaging one that usually gets formed first and as actually then converted, its product is converted by catalase? Right, superoxide demutase is the first one that it usually forms, and superoxide demutase, that's the enzyme that converts it into hydrogen peroxide. But we do also form directly hydrogen peroxide. So all of our organisms bubbled, right? Because all of our organisms are utilizing oxygen. We know that because of what test we ran. The FTM, right? Some of you guys have facultatives that can grow anaerobically, but it could grow aerobically with oxygen. It can utilize an electron transport chain with oxygen as a final electron acceptor. So it makes sense that all of ours bubbles. If you looked on that list, you guys could figure out which one was my aerotolerant. Because it's the only catalase negative I had on that list. Right? Because does it need catalase? No, because it's not doing aerobic respiration. Right? It's probably doing fermentation. It can grow in the presence of oxygen because it does have superoxide demutase, but we don't have a test for that. We just have a test for catalase with hydrogen peroxide. But because it doesn't have an electron transport chain that it's utilizing, it doesn't produce as much toxic forms of oxygen or is exposed to them, so it doesn't need to have catalase. So the methyl red test is for mixed acid fermentation, right? And this is using the pH indicator methyl red that let us know when we added it that the pH was 4.4 or below, right? It stayed red. Note that it turned what color if your organism was negative? It turned yellow, right? So it seemed like it disappeared, right? Because the media is also yellow, right? But it doesn't, in fact, truly disappear. It just turns the same color as the media. So we asked you some questions in your report, right, relating this test to phenol red. And I tried to draw on the board before to explain this, but I put together a very fancy slide yesterday. And I thought I lost it last night. I was so depressed. I was like, where did it save on my computer? I know I hit save. I found it. So I put a lot of work into this. So this is the pH scale, right? So from 0, right, to right about 7 is what we call acidic. 7 is neutral, and 7 and above is alkaline conditions. So we said that methyl red stays red, which is a positive result for mixed acid fermentation, at 4.4 and below. So I'll color this in from 4.4 below, right? If your organism measured in this range, it would turn red, right? What about for phenol red? Is it the same range? No. What color does phenol red, that pH indicator, turn when you have acidic products produced? 6.2 and 6.8 and below. Right? So from here down. So the question asks, would you suspect a methyl red positive organism to be phenol red? Yeah, see, these two overlap, right? If it's anywhere here and below, it's also going to turn yellow, right? So if you have one, right, say we tested the pH, it's 3.5. That's going to be positive for methyl red and phenol red acid production, right? Make sense? So the answer to that one's yes. Will the reverse be true? If an organism that is phenol red, will it be methyl red positive? Not necessarily, right? The answer is no on this one. So can you guys think of where the pH could be that it would be positive for phenol red and not methyl red? Right? It's got to be greater than 4.5. So something in this range of, say, 5.5, right, is going to be phenol red positive, but 5.5 is above 4.5, right? It's not going to turn methyl red positive. So the answer to that one is no. One of the other questions is, can a methyl red negative organism cause the pH of the MR.VP culture media to drop at all from its initial value? So the first question is, what's its initial value, right? So its initial value starts out in this range here of 6.7 to 7.1. So could it go below this and not turn red? Sure, that's some of those organisms that are phenol red positive but not methyl red, right? It could be 5.5, right? But it's got to be 4.4 or low to end up being positive. So unless you actually test the pH, which we really don't need to do because, right, we do both of these tests that test for acid production. This one for obviously a lot of acid production, right? Usually the organism produces a mixture of acids, hence why it's called the mixed acid fermentation test. Okay, and with phenol red, we tested for glucose, but there's lots of different sugars you could test. But again, that's a reason why we do glucose, because that's the same sugar that's in the methyl red test, is glucose. Right? We couldn't compare these two if we were using a different sugar in the phenol red broth. But we're using the same sugar, glucose. So the answer is yes. Okay? Did these make better sense to you guys now with the pretty pictures? Good. It was worth my effort and time. Okay, so the Voges-Proskauer, remember, detects this intermediate, acetoin, which can be turned into 2,3-butanol, right? This is the only fermentation pathway we're actually testing for an end product that is a what? What is 2,3-butanol? Is it an acid, an alcohol, or a gas? It's an alcohol. Remember, alcohols end in OL, right? And this one, just like methyl red, which we did both of these from that same broth tube, right? We took aliquots, and then, but this one was a chemical reaction that needed to happen, right? We had to add the reagents in order to form the red compound to get a positive result. Remember, we had to wait as long as 60 minutes, potentially, for that red color. Though most of ours didn't take much time at all. So then what we have left is our selective and differential media. Remember, if we're selecting, there's something toxic in that media that's stopping the growth of one group of organisms and therefore selecting for the group that we care about, right? We're inhibiting the ones we don't like with the selective agent and allowing, therefore, the ones that we are selecting for to grow. So the Colistin was an antibiotic found in which plates? Yeah, the CNA blood agar plates. The CNA, C stands for Colistin, right? The other was nalidixic acid. So the antibiotic is a selective pressure, but sometimes the simple compound sodium chloride we know is salt can be a selective pressure. Which plate was that? The MSA, because it stands for Mannitol Salt Agar. And in that case, the sugar mannitol is in its alcohol form. Sugars can actually be alcohols too. If they can't survive on a particular type of selective media, then you can't say anything about the differential aspects of that bacteria, right? I think I pounded, hopefully, that one in pretty well on you guys. If you don't have any growth on those plates, right, you couldn't differentiate which group they fall into for the next classification, right? They have to actually grow. So if it was selected for, if it was removed, right, you can't get any other information.
So in our explanations, do we need to go into further detail, or if it's just that you if you ended up with a negative?
Yeah, you still need to explain, right? For those, you still need to explain that had it grown, right, you know what yellow means, right, and what the different hemolysis look like. But I think those I have you guys explain separately for those, right, not for your organism. You answer those questions. I won't be as particular on the plate media as I am on the tube media in the explanations. Okay, so mannitol salt, select for salt tolerant, right? So only salt tolerant grows. So if yours didn't grow, it's salt intolerant. If it is salt tolerant, then you could ask the next question, does it ferment mannitol? And typically why we use these plates, we're looking for what organism that ferments mannitol. We're usually looking for Staph aureus, right? But that would be in a clinical lab specimen, right? And we may have some contamination of Staph aureus in this lab. I haven't tested it yet. There's another test I can run. But we had a lot of yellow plates, so I don't know what's quite going on there. But Staph aureus, most of us carry around in our nose, so it very easily contaminates cultures. Yeah, your organism may be doing that, or it may be contaminated. Staph epi doesn't, so it's probably a contaminant. Right, I think we've got some contamination going on. Okay, so EMB, eosin methylene blue, are the dyes, right? And remember, these inhibit what group of organisms? What won't grow on these plates? Gram-positive. So if you have growth, it confirms yours is gram-negative. And then depending on the colors or lack thereof color, tells you the next step, the differential part, does it ferment lactose? Because remember, the formula we have just contains lactose for these plates. So if it kind of blends in with the plate, right, it's not a fermenter. If it's pink or purple like you see here, it's a moderate fermenter. If it's this pretty metallic green sheen or black, right, this is a characteristic of organisms like E. coli that are really strong fermenters of lactose. And it's the acids that they produce that react with the dyes that cause these color changes, the eosin and the methylene blue in the media. So for Columbia CNA plates, right, these contain the antibiotics, Colistin and nalidixic acid. These inhibit the growth of gram-negative. So who grows on these plates? Just gram-positive. So only for the gram-positives could we ask the question if it was hemolytic. And there's two types of hemolysis that actually lyse the red blood cells. Beta, and what happens to those red blood cells when it's beta hemolysis? Completely lyse. So in the case of this one where the bacteria ran out of food and started forming spores, so you don't see the growth anymore, you can see right through that plate, right? Where the alpha has what kind of colorization to it? It's kind of greenish, right? And this is incomplete lysis, where they poke holes and then the oxygen goes in and decolorizes it. Gamma, is that hemolysis of the red blood cells? No, it just means it grew on the plate, right? And in this case, we would then know it's a gram-positive, which is the case for some of you guys. So justification of inconclusive results is important, right? Do not, right, if you got, say, a negative result for citrate, just per example, right, in your report, on that last page, you need to write negative. But if for your organism it was supposed to be positive, you need to put a little star on it in that section where it says justify, justify. I think everybody, though, didn't have too many inconsistent results where they couldn't narrow it down, though, to their organism, right? If anybody's not sure, come see me, right? But make sure that you explain what could have potentially gone wrong, right, with that particular test that's not matching up. Don't fabricate results so that they match the table, right? Do your explanations. I don't think I need to read the rest of this to you guys. Okay, so this will be posted for you along with the pictures. So let's get to the fun stuff, which I didn't open yet. Is the final exam review. So as it says on the paper you have, right, I don't think I need to read it word for word for you guys, but reread your syllabus. That's a good, like, you know, what should I know, right? We put a lot of work into that document. Go over your homeworks, go over your quizzes, right, from Lab 7 on. Right, you don't have to worry too much about the stuff before then. Your graded unknown lab report would be an excellent study tool, except you guys aren't going to have it, right, because I let you guys turn it in today instead of last week. But I will be grading them over Thanksgiving holiday. I will post messages to you guys, right, if something's really off. And that's, again, why you need to get your explanations in by the 22nd, so I can give you guys feedback, right? So make sure you look for that. But just print out a blank copy of the unknown report, right? See if you can answer those questions without looking at your notes, right? It itself can be used as a good quiz to help you review. The PowerPoints, especially those scientific method slides, are great. Create your own tables of information. Try and summarize information. See how much you know and remember. So the exam covers all exercises after midterm, right? So 7 on. Most of them had to do with your unknown. That's why the unknown report is a really good place, right, for studying. All of these tests, right? There's 12 different tests that you'll have to interpret again, right, on the exam that you've already done on your unknown. Standalones that you need to not forget about that we went over is that standard plate count, so redo that bonus quiz, right? Antibiotic sensitivity, that's pretty easy, right? Review your quiz for that. Transformation, which we did last week, and I'll grade your quiz and give it back to you on that. Hopefully you have a pretty good understanding. And then we're going to do ELISA today. And as I said, I'm going to have a bonus quiz for you guys on that. So to give you guys a little more practice, because you've only really done this once, right, is being able to process of elimination down and figure out what an organism is based on results, right? So, right, you'll evaluate stains. We'll do that separately, right, so that you're not doing double jeopardy, right? If you look at the microscope and you don't identify the right thing, then I ask you another question based on that. So it'll be just like this handout, where I'm going to give you the table, right? By no means are you memorizing that table of known results. You're going to get a copy. You can write all over it. It'll be yours, okay? And then we're going to give you test results just like here. So say a student has obtained the following result on his or own unknown organism. Natrell, is there a result on here that kind of jumps out at you? That's kind of rare. Phenol red as being K, right? We really didn't see any pink tubes, right? So that one's kind of rare on our list. What's even rarer than that? The acid fast. We know it's probably the mycobacteria, right, on that list. Just with that result, but you would confirm going through the rest of them, right? But this would be the Mycobacteria smegmatis, right? So if we walk through the results, right, acid fast, positive, you look at that chain, look, is the only one on this list, right? That's not always going to be the case, but again, you would have very limited possibilities with that one result being positive, right? So what about the second one? A student has obtained the following results on his or her own unknown. So gram positive, right? That divides our possibilities in half, right? A acid with no gas for the phenol red. Motility negative. We didn't have too many of those, huh? Capsule positive, catalase positive, and citrate negative. What did you narrow this down to? This actually brought you to 2. Which 2? Right, the 2 staff, right? So, right, get rid of all the negatives. We're left with the positives. Phenol red, got rid of the K's, right? Left these possibilities. with the motility, it eliminated all the bacillus, right, and the citrate test. So we're down to the 2 staph and Enterococcus facialis, right? We said the catalase was positive, so that eliminates Enterococcus facialis and left us with these 2 guys. So what test could we run where these guys apparently are different? We could run the VP, right? Right? You see, that one's different. This is a test you could run, right? VP test. Make sense? Okay. Are you going to rely on just one test? What if something goes wrong, right? You want a second test to back that up. So in this case, what plate media would you use? You could use the MSA, right? That would be the most common one to use because what do you know is going to happen as far as Staph aureus or Staph epi in the MSA plates? They're both going to grow, but they're going to change the media, right? Which one's going to change the media to yellow? The Staph aureus should turn it yellow, right? So that's one thing you could have done. One of the things you could look, if you look at your unknown report, which I would not ask you guys on the exam, so I don't expect you guys to memorize this, Staph aureus is beta hemolytic. So what would that plate look like for our blood agar? You'd be able to see through it, right? You'd see a zone of clearing, right, where it's completely lysing the red blood cells. Where Staph epi usually does not do that. All right, so let's do another one. Gram negative, it's rod, acid no gas, phenol red test positive, capsule positive, and citrate positive. All right, so let's see what one does that narrow down to. Serratia marcescens. Okay. Right, so gram negative eliminates all the positives, right? Half our chart gone. Phenol red, positive, we got rid of 2 of them. Citrate, positive, brought us down to Klebsiella pneumoniae and Serratia marcescens. If you look up, remember Klebsiella pneumoniae is pathogen, right? So nobody got that one as a result, right? We did have someone who had this organism, right? And we found out recently that this one, if you re-incubate it, it will actually eat the protein. So this one ends up with a weak K. Yeah. Under certain circumstances, it can be pathogenic. Okay. Oop. I gave you the answer to that one already. So this one, with it being spore positive, we can narrow it down to the 3 bacilli. And then they give you one more test result, and we narrow it down to which 2 bacilli. Yeah, subtilis. It's actually subtilis. Like subtle, but it's not subtle, you know? Megatherium. Okay, so what test from the chart? So we end up with spores, right? So we narrow it down to 3, right? Phenol red, we narrow it down to these 2, and the anaerobic growth. So what test is different for these guys? VP. Again, are you going to rely just on VP? No, right? On the exam, again, I'm just going to ask you what test, right? You're going to narrow it down to 2. What test? I'm not going to ask you about plate media. But what could we use for plate media to back this up? We could use the blood agar because, again, if you look at your unknown report, one of them is beta hemolytic, the other is not, right? So the Bacillus subtilis, oops, what happened? Oh, there we go. Would be beta hemolytic, and the other would not. So for some of you guys that had those bacilli, right, that should have hopefully helped confirm. Though I know we had some growth issues. So the rest of this I'm not going to go over, but I'll make available to you guys, right? Just a general review of the information in the course.